Sweet. Okay. We're recording. So last week we did protostomes. This week we're doing deuterosomes. These are all the triploblasts, if you remember. So that's three germ cell layers, right? Last week we did the protostomes, which include the mollusks and the annelids and the arthropods. This week we are doing echinoderms and us, things with backbones, the chordates. The difference between the protostomes and the deuterosomes, it's all encompassed in their development. Okay? So we'll get into that in a little bit. Just these slides are really important. The slides that show the family tree in all the animals, because they have all these dots that tell you the defining features of those groups, and that's going to be super important. In the animal lab, or the animal labs, the focus of your learning is going to be on knowing morphology and knowing taxonomy and how those two tie together. So we'll start from the beginning. We have the bacteria. The important thing to know there, lab techniques, right? In the plants, the important thing to know was life cycles. Here in the animals, the most important thing to know is how these different body structures and how the different structures within the body structures function to make these guys more and more advanced. Okay, so that's real important. So for the echinoderms and the hemichordates, this group up here, ciliated larvae are some of their defining features. Since you don't need to know the acorn worms, the hemichordates, we're going to treat this entire clade as just the echinoderms. Those are the only ones that you're required to know. So the things that set them apart from the rest of the deuterosomes, number one, they have radial symmetry as adults. So all these guys, if you recall previously, all the triploblasts that we learned about are under that group called the bilateria, which means they have what? Bilateral symmetry, front to back symmetry, right? That's hard to reverse because bilateral symmetry is so useful as an organism. But if you remember, if you're a kind of like a lazy ass animal that doesn't like move around a whole lot, like a starfish, like an urchin, you don't need to have that. So when they grow up, when they're young, they look like our embryos, kind of, sort of. They have very similar looking larvae when they're young. But as they grow up, they turn into radially symmetric organisms. So that's something to know. They also have calcified internal plates, so their endoskeleton. If you brush your, I don't know, if you've ever seen like a sea star skeleton, that's all calcium carbonate. That's like right underneath the skin. So they have this like kind of chainmail skeleton inside of a calcium carbonate. Also, they don't have pharyngeal slits. So when they're young, and when we're young, all these chordates, they have these like gill-like things, these pharyngeal slits. They act as gills in some of these chordates. They eventually turn into our jaws and other structures in the inner ear, but these guys don't have that slit at all. Okay. So we'll go down to the chordates, which are the most advanced, quote unquote, animals that you'll be looking at, and these are the ones that we're a part of, right? The vertebrates are within this group. All of these guys have something called a notochord, which is, so the things that really set the chordates apart, they're like very well delineated. They're on your lab notebook, they're on the slides, they're everywhere. Those are the kinds of things, when you see those bullet points of like 5 or 6 things that tie a group together, make sure you tailor your studying for that kind of shit, okay? Okay, so the couple of things that tie all the chordates together, one, they have notochord. So you guys remember what a notochord is? Anyone have an inkling what that is? You've done your pre-labs? Allie? Yeah, so it's just I forgot to go and do the directionality in the previous lectures, but so when you have an animal, right, like this, it's bilaterally symmetric, you have dorsal, which is your back, ventral, which is your stomach, anterior, which is your front, and posterior, which is your back. I'll put that in the notes, but that's something that I didn't go over last week, which I should have. Okay, but yeah, so it's this cord, this piece of connective tissue, which spans the dorsal side of all chordates, right? In the basal chordates, that basically serves as a backbone. It is a non-skeletal structure that acts as support for the back of the organism, right? For us, as you get more and more advanced and you actually have a skeleton, that notochord actually becomes the discs within our spine, right? So bottom line is the notochord serves as a tethering and a structural support for the back of the organism, quote unquote back, right? Also, you'll have a nerve cord in that same area. Dorsal hollow nerve cord. And that is, of course, a collection of neurons, acts as a centralized highway to link all the neurons from across the body back to the brain of the organism, okay? And finally, you have a post-anal tail. So, how do I draw? I'll draw this out. Sorry, the iPad's malfunctioning. So, the post-anal tail, so you'll have this organism like this. You'll have the notochord, which disappears in some groups, or becomes other things in some groups, but it stays in others. But at some point in time in development, they have a notochord. And then you have a nerve cord right underneath, which links the brain to the rest of the body. And so you have a post-anal tail, which actually, it's the extension of this notochord all the way downwards, like that. This is the butt right here. What this means is that you have a tail that's not linked to your butt, so when you move around, when you're moving like rapidly, you're not moving your GI tract necessarily at the same time, right? For example, like something that doesn't have a post-anal tail, that GI tract will go all the way to the end. So when it's moving its tail around back and forth, it's actually moving its gut, and clearly, like, that's not necessarily that efficient. Okay. So those are notochords: dorsal hollow nerve cord, post-anal tail, and they have pharyngeal gill slits. So these guys lost them, the echinoderms lost them, but the chordates, just remember notochord, pharyngeal gill slit, post-anal tail, and nerve cord. Okay? Next, we move on to the true vertebrates. These are us. Now they actually have a skeleton reinforcing that notochord. So the notochord becomes the discs within the back of that organism, right? So you have a spine, which is actually skeleton. Each disc in between is that connective tissue that originally made up the notochord. They also have a skull, so an actual case for the brain, which is skeletalized, large brain, and they have 2 to 3 chambered ventral heart. Pretty self-explanatory. Okay. Blah, blah, blah. Okay, so the difference between the protostomes and the deuterostomes that we're going to go over, it's all related to their development from an embryo. If you remember in the protostomes, as they go from 4 cells to 8 cells, so you have 1 cell, that's the fertilized embryo, divides into 2, divides into 4, and once you divide into 4, you need to divide upwards, right? In the protostomes, if you recall, when they divide upward every single time, the cells shift a little bit, and so you actually get a spiral pattern upwards. Maybe not that visible here. In the slides later on, it'll be more obvious, but that's called spiral cleavage. As you grow and grow and grow, the embryo grows upwards in a spiral fashion, right? And how you'll be able to tell is, so right here. The spiral cleavage, that second level of cells, the fission barriers right here, each cleavage site, from the second level to the first level, they're not overlapping, right? Whereas in radial, they're overlapping. You see that? So that's just a memorizational thing. You don't need to know why that is. I don't know really why that is, but it's important to know that that's one of the differences between the protostomes and the deuterostomes. Next, as these embryos grow and grow and grow, they turn into this hollow ball like this, right? That's called the blastula. That next step is to take a part of this blastula and punch it inwards to make like this bowl, right? The opening of that bowl is called the blastopore. In the blastopore in the protostomes, that becomes the mouth. Whereas in the deuterostomes, that pore becomes the anus. Again, just a developmental thing. These are all just pure memorization. I can't really help you with that. You just gotta know. Okay. And then finally, at the 8 cell stage right here, in the deuterostomes, in the protostomes, remember, if you take one of those cells away, that entire embryo dies. Because each one of those embryos, each one of those cells has a fate. It's going to become a very specific set of cells in the adult organism, right? So that's called determinate cleavage. In the deuterostomes, you can take each one of these 8 cells and pop it off and throw it in a test tube and it'll grow into a new embryo. So each one of those 8 cells does not yet have a determinate fate, so it's indeterminate cleavage. Okay, so just backtrack 3 things: differentiating deuterostomes from protostomes, spiral cleavage versus radial cleavage, determinate versus indeterminate cleavage, and the blastopore being either a mouth or an anus. Okay, again, these are the kinds of things, these little differentiations that are often memorization heavy. You got to know. I'll put up another study sheet on like those specific differences that you'll probably need to know. I don't guarantee they'll cover everything, just like before, but really it's just up to you to go through the slides and just grind it out. I'm sorry. Okay, so synapomorphies. So all these guys are triploblastic. We already talked about that. They have a coelom. They're truly coelomate. Let's see. Bilateral symmetry. Pharyngeal slits. Remember the opening between the pharynx and the throat. It extends your internal environment to the external environment. When you're young, those tend to be, in those guys that keep those slits, they become gills. All right. At least in the basal chordates. Do you guys remember what a coelom is? This is going to be something that's real important. What's a coelom? It's a body cavity, right? It's filled with fluid. What is the point of having a coelom? This body cavity. Why do you have a body cavity? 2 things that we talked about. Yeah, it protects all your internal organs. Yeah, right, exactly. It's like an airbag. It's a water balloon surrounding your entire body. If you smack the outside of your skin, all that pressure will be diffused by all the water inside. What was the second thing that we talked about? Coelom. What was the purpose of coelom? No. Besides impact. It's the same thing. So the second thing is a little less obvious. When you have a watery environment inside, it's filled with water, right? It's a fluid-filled cavity. It allows you to disperse gas a lot faster, dissolve gas a lot faster. So you oxygenate your blood a lot more efficiently. Okay? Cool. And what distinguishes a pseudocoelom from a true coelom?
Mesoderm being on the outside of the gut, on the inside.
Right. So in both, they have a mesoderm surrounding the inside of the ectoderm, but only in the true coelomate organisms do you have mesoderm surrounding the endoderm. Okay, that's super important. That's something you'll for sure have to know. Why? Yeah, we'll go over that in the review. All right. Okay, so just coelomate. Very, very important. Remember that concept of the coelom. Like, that's one of those central things that really, it's one synapomorphy that has its own slide, which means you got to know that's real important. All right, so we'll move on. So we're on the echinoderms now. Echinoderms are sea cucumbers, the ones that you need to know: sea cucumbers, sea stars, brittle stars, and urchins. Okay. Like I said before, it will be important this time around to know scientific terminology. So you want to know, echinoids, ophirotheriids, asteroids, holotheriids. These are like important. There's a reason you have to know these like names, right? They don't just want you to memorize these things willy-nilly, but for things like classes and phyla, you'll want to know the actual scientific name for that file or that class or so on and so forth. Okay, so these guys, like we said, most of them as they age into adulthood, they have radial symmetry, except the sea cucumbers, right? And a large majority of these guys, how they feed, they either filter feed or they crawl around the seafloor eating random bullshit, right? Some of the starfish tend to be more predatory. They like to eat clams and like dead fish and stuff like that. Ophiurids, the brittle stars, they tend to eat like more dirt. Same with the sea cucumbers and the echinoids, the urchins, they really like to eat algae. All right, so let's go over the basic overarching themes for the echinoderms. Number one, they have a complete gut. So they have a real digestive system, even though it's not particularly advanced. Number two, they have a water vascular system. So again, another one of those things that has been talked about a bunch of times, so you want to know. The hydrostatic skeleton is what this kind of classifies itself as. What is a hydrostatic skeleton again, and how does it work?
You squeeze one side.
Yep. It's moving around water inside of a confined space. It allows you to extend your features or retract them, right? Just based on the movement of the water. It also provides support, right? Water provides some amount of support. In the case of these guys, if you'll see this as you look around the actual organisms, they have these things called tube feet. And if you've ever seen the underside of a living, actually, let me pull it up online. It's going to be important to know. How do I escape? So they have these tube feet. This is all these feet are connected to each other through one centralized repository of water. That's their hydrostatic skeleton. And they can move that water in and out of that tube feet to move those tube feet, put them onto prey, retract the prey. It's how they move around, basically. Okay, so they'll move around on the ground like this, and the entire process of moving involves the cumulative action of moving these tiny little tube feet. Okay? And so that's linked to their hydrostatic skeleton. Play from current slide. Okay. And they have a huge coelom. If you actually see the cut open starfish, most of their body cavity is coelom. They're marine. They're all marine. They have pental radial symmetry, which means they have radial symmetry, but it goes in 5 planes. You can clearly see that from the starfish, right? And they have a calcareous endoskeleton. Remember, the endoskeleton is made of calcium carbonate. So those are things you got to know. Clear differentiation between these groups, right? If you want to look at like how they look, the brittle stars have a centralized body in here and really, really long, tiny arms going outwards, right? The urchins, you know what urchins look like. Kind of a round, bulbous, centralized skeleton with spines sticking out. The tube feet are underneath. There aren't tentacles moving outwards. Starfish, they look kind of like brittle stars, but they're much meatier, and they don't have a defined middle region that's sectioned off like the ophiurids. The sea stars, it's just one muscly mass. Okay, and then the sea cucumbers are the only group that has bilateral symmetry. and that's all you really need to know in terms of how they look. So again, it's always important to be able to identify the different groups. Okay. So be able to identify the 4 groups. Like you said, you want to look at the sea star dissection down there, identify the features, and then look at you have slides in front of you that have the early development of these echinoderms. All right, so let's take a look at the inside of these echinoderms. We're going to focus mostly on the sea star. It'll be in your lab notebook too, so just follow along. We'll start out with this top thing right here. So, important to know more than anything else, you have this on the top of the sea star, which is actually called the arboreal. So because of their interesting body breakdown, they don't have anterior, posterior, dorsal, ventral. They have either the plane or their side where their mouth is, which is the bottom, which is called the oral side, or the arboreal side, which is the top. Okay, so on the arboreal side, the top of the echinoderm, they have is one pore right here, which is like right here. That's called the madreporite. The madreporite acts as the pore which either sucks in or releases water into their entire skeleton. Their hydrostatic skeleton, the amount of water that is inside that hydrostatic skeleton is regulated by the madreporite. Okay? Okay. That's below. So we'll talk about the hydrostatic skeleton a little bit later, but the number one thing you want to look at, top to bottom, the first thing you want to look at is the digestive system. So you have the mouth down here. The mouth brings food in to their stomachs. So their first stomach, down below the cardiac stomach, which is this fat cushion down here, that takes in the food immediately from the outside. The way these echinoderms tend to eat is they like to throw up their guts, right, to their external environment and kind of digest the whatever it's been eating. Throughout their guts, they release digestive juices and they take everything back in, right? What happens then is that first stomach will take in all the guts, will take in all that food, which has been pre-digested a little bit, and grind it up. So that's cardiac stomach. Kind of looks like a heart and it beats around, so cardiac. All right, and then above that, that food then gets transferred into the pyloric stomach, which is up here, this star-shaped thing, and it gets fed a bunch of enzymes and digestive material from the digestive glands, and it'll continue to digest that food. It'll eventually come out the top through another pore, which is the anus. So at the top of the starfish, there are two pores. One, which is the madreporite, important to know that term, which regulates water, and then another one, which is the anus, which throws out poop. Below all that is the hydrostatic skeleton. So, yeah, stomach, so pyloric stomach, cardiac stomach, and then below all that is a ring, like a ring of tubes that is connected to the outside with madreporite. So I'm going to draw that out here. So the hydrostatic skeleton looks like this from the starfish. You have the madreporite, which is the pore that connects the outside to the inside. Then you have a ring canal, and then from each one of those ring canals, you have a radial canal, or two radial canals that feed water into these little sacs called ampullae. And each one of these ampullae is connected to the outside with a tube foot. Okay, so if you want to track the flow of water, you get water in from the madreporite. That water will go into the ring canal, and then it'll spread from the ring canal into each one of the feet through the radial canals, and through that radial canal, it'll end up in these little sacs called ampullae, which then squirt the water into the tube feet to allow the tube feet to move. Okay? Alrighty. So we went over all that stuff right here. This is the actual dissection of the starfish. You can see it right behind Liana. The only thing I didn't mention are the gonads, which are right below the digestive tract. Okay. So the gonads, all their internal reproductive organs are right below their digestive organs in each one of the arms.
John,
Yeah.
can you go back to the previous slide?
Yeah.
Or like second to last.
This one?
Yeah.
So-
With the purple diagram on the top right, I got confused with the diagram. Do only one of the arms have gonads, or
No, all of them have gonads. Yeah. So it goes.
it's just different layers showing the arms?
Yeah. So from the bottom up, the bottom part of the very bottom of the arm is the radial canal with the ampullae. The next layer up is the gonads, and the next layer up on top of the gonads is the digestive glands.
Thank you.
Yep. Good question. All right. And so this is actually not what you'll be seeing. What are in your slides are the growth of a sea star embryo, which looks reasonably similar. But what I want you to focus on is that as you grow up, you actually get this bilaterally symmetric embryo that eventually turns into a radial symmetric organism, adult. So let's break for 5 minutes. Take a look at all the echinoderms that are in that box. 5 minutes. Look at all the displays that are on there, both the broken up starfish and the model, and then we'll answer the questions together at 9:40.